The generic formulation development is based on the reference formulation. The reference formulation for the US market is called as reference listed drug, or it is also called as the reference standard. All the reference standards are RLD, but all the RLDs are not reference standards. Reference standard is the R&D strength, the reference listed drug strength, which is used for the bioequivalence studies or for the in vivo studies. As the generic formulation is required to be equivalent to the reference formulation, selecting the RLD becomes very much important. This video is for selecting the RLD RS for a US ANDA. US ANDA, that is US Abbreviated New Drug Application, is a generic application for the US market. And for filing the ANDA, proper RLD selection, RS selection is required, so that you can reference that formulation in your application and you can file based on that. I welcome all the viewers to this topic on selecting the reference formulation for US ANDA. Selecting the RLD is very much important, as the RLD is used for referencing the generic formulation. Generic formulation is developed inline to the reference formulation. Reference formulation for the US is called as reference listed drug, and the generic formulation is required to be same or should match the RLD in dosage form, strength, route of administration, labeling, quality, and the bioequivalence. So all the quality related, safety related, and efficacy related things should meet the RLD. So whenever you are developing a formulation or generic application, that is ANDA for US market, where you will find the RLD. So the official source is FDA's Orange Book, and in this Orange Book you can search by active ingredient, proprietary name, applicant name, dosage form, route. So these things you can try and search. Then in the Orange Book, FDA designates one product per strength per dosage form as the RLD. So if you are working onto a formulation like tablet, you should check the tablet formulation. So your dosage form, your strength, route of administration, labeling, quality, all should be in line to the RLD. Then step 1 for the selection of RLD. Confirm if an RLD is listed. So look under the RLD column. The RLD column will have plus symbol or RLD tag. If present, this is the mandatory reference for your ANDA. You cannot use any formulation as a reference. The US FDA designates the reference formulation. Step 2, if RLD is not there or if no RLD exists, then sometimes the RLD is discontinued or not assigned. FDA may allow you to select RS. RS is a reference standard, the product used for the bioequivalence testing. If RLD is not available, RS may serve as the reference. For example, if RLD is discontinued, then FDA will provide you the reference standard to perform the bioequivalence studies. RS may serve as a reference for the formulation, BE study, and quality comparison. If RLD has many strengths, then RLD and RS both designations will be there, and you need to select RS for the bioequivalence studies. Then step 3, if both RLD and RS exist, that means you need to use RLD and RS both. RS is used for the in vivo BE studies and in vitro studies like dissolution and IVIVC. So this is very common for the US products. If RLD is having three strengths, for example 10 mg, 20 mg and 50 mg, and if the 50 mg strength is used for the bioequivalence, then 50 mg strength will be designated as RS. RLD is used for the formulation Q1, Q2, Q3 comparison, labeling match, and the development targets. RLD versus RS. So RLD purpose is basis for labeling and formulation sameness. RS is basis for bioequivalence. RLD and RS both are selected by the FDA, and RLD is used when it is available, and RS when RLD is unavailable or FDA specifies. Sometimes both are available. That time RS is used for the bioequivalence studies, in vitro studies, in vivo studies, and RLD for the formulation sameness. Key criteria for selection of the RLD. The selected product must match dosage form, strength, route of administration, and the release type. You cannot mix different release mechanisms or strengths across the RLD. Important regulatory points for the RLD and RS selection. If RLD is discontinued, it can still be used as RLD as long as it remains in the Orange Book, but it should be available in the market. If multiple brands are there, then FDA still selects one specific RLD. You cannot choose any other RLD than this. Then authorized generics cannot be used as RLD. Reformulated RLD. If innovator reformulates, old version may become discontinued, new versions may become RLD. So you need to check all this information very carefully. Practical example, scenario 1, RLD is available. Use that for Q1, Q2, Q3 sameness, labeling and target dissolution. Scenario 2 is RLD is not available. So use RS for B, but you may need justification FDA correspondence, that is called as control correspondence or CC. Scenario 3, RLD discontinued but still listed. So still acceptable by the ANDA. Then some tips are there. So always check the updated Orange Book. It get updated on a regular basis. Record the screenshots for the documentation and you will have the ready reference. Submit pre-ANDA controlled correspondence if RLD RS is unclear. You can ask FDA through control correspondence and they can guide you. Then FDA may issue guidance recommending specific RLD RS. Also, always check the product specific guidance for BE studies. In that, you will understand the strength on which biostudy is required to be performed and the strengths which are waiver strengths. So, RLD is a regulatory benchmark for labeling and formulation. RS is benchmark for BE testing. Always study and understand the RLD labeling. Always visit the Orange Book and understand the RLD and RS. So this is the information for selection of the RLD and RS for the US market generic application, that is ANDA. Thank you for watching the video.